Welcome friends, welcome in another video lecture of Easy Life Science. Here I am talking about Endospore Staining by Dorner's method. So please subscribe my channel Easy Life Science, like my video, share it to your friends and don't forget to press the bell icon so you will get a notification of newly uploaded video. So let's start the history of endospore. The endospores were first described by a scientist Kohn in 1872 in an organism Bacillus subtilis and later by a scientist Koch in 1876 in the pathogen Bacillus anthracis. The Kohn demonstrated the heat resistance of a endospore in a Bacillus subtilis, and a Koch described the developmental cycle of a spore formation in a Bacillus anthracis. In 1922, the scientist Dorner published a method for a staining of an endospore, and this method was so lengthy and having a heating step, but resulted in a differential staining of a endospore and a vegetative cells in a same sample. In 1933, scientists Schaeffler and Fulton modified Dorner's method and made the process faster. The endospores are named because they are formed intracellularly, although they are eventually released from this mother cell or a sporangium as a pre-spores. An endospore is a spherical or oval, thick-walled, highly resistant structure formed in a certain bacterial species that represent a dormant or a resting stage in a growth cycle of an organism. The organism which forms an endospore are called as a spore forming organism. Example, Bacillus species and a Clostridium species. The formation of endospore in a cell is called as a sporulation, and the sporulation occurs at a suboptimal conditions or a diverse condition of environment or growth, like exhaustion of nutrients in a medium like a carbon and a nitrogen sources, a high temperature or a low temperature. The sporulation is not a method of reproduction. The typical structure of endospore shows a wall that is called as a spore wall, and this is a delicate membrane from which the cell wall of a future vegetative bacterium develop. Spore cortex, spore coat, which is tough and multilayered, and exosporium. These are the part of the endospore, and this is a image of a typical endospore. The endospores are resistant to UV radiations, osmosis, desiccation, high temperature, low temperature, against a chemical disinfectant, and at time of starvation they are resistant to these conditions and they are destroyed by autoclaving and thinalization. When a spore get germinated into a vegetative cell, this is called as a germination, and the germination of spore, it occurs at optimum conditions of environment and growth, and it leads to an increase in metabolic activity of a spore. So because of a metabolic activity, the endospore gets swelled and a germ cell appears by a rupture or a absorption of a spore coat, and then this germ cell elongate to a vegetative bacterium. Shape, position and size of a spore relative to a parent cell or a mother cell. The shape of a spore may be bacillary body. Example of such spore in a Clostridium species do not having a such like bacillary body. Example, Bacillus anthracis. The position, it may be a central. Bacillus anthracis, Bacillus bifermentans. These organisms having a spore which is at a central of a cell. Terminal, Clostridium tetani and Clostridium tertium. Subterminal, Clostridium perfringens. Shape may be a oval. For example, Bacillus anthracis, Clostridium tertium. It looks like a tennis racket. The shape may be a spherical. For example, Clostridium tetani. In this image, there is a spore in a Clostridium species found in a pus sample. Here in this slide, there is a list of spore forming bacteria. These are Bacillus species, Clostridium species, Thermobacillus, Thermomicrobium, Sporolactobacillus, Geobacillus, Geosporobacter, Oxobacter, Acetonema, it is a gram-negative bacilli, Porosarcina, it is a gram-positive cocci. And these are some examples of spore-forming organisms. Then the methods of a demonstration of spores. The spores can be observed under a phase contrast microscopy. In a phase contrast microscopy, the endospores are observed in unstained wet films. And during observation, they appear as a large retractile oval or spherical shaped within a bacterial mother cell or elsewhere free from a bacteria. And this is an image which shows a spore by a phase contrast microscopy. And this is a spore present in organism Bacillus thuringiensis. The method given by a Dorner's for endospore staining, requirement for the procedure, requirement for this method. First requirement, that is a 24 to 48 hours old cultural suspension of Bacillus species, Gill Nielsen carbol solution, 10% aqueous nigrosin solution, a small test tube and a water bath. Procedure: place a few drops of Genesep in a test tube containing a 24 to 48 hours old cultural suspension of a Bacillus species. Then place this tube in a boiling water bath for 20 minutes. During this treatment in a boiling water bath, the Genesep contains a stain, that is a basic portion. It will get absorbed by a endospore. Here the steam acts as a physical intensifier and a carbol portion in a Genesep acts as a chemical intensifier. So during this treatment in a boiling water bath, the vegetative cell as well as endospore get colored, and it takes a color of a basic portion in a Genesep. Then third point, remove the tube from a boiling water bath, cool it, and take a loopful suspension from that tube onto a clean glass slide, prepare smear of it and heat fix. After this, place a few drops of 10% nigrosin at one end of a slide and spread it over the smear with the help of another slide. Then allow a smear to dry and do not wash the slide at this time. 6 point, After it, examine this line under oil immersion lens. Observe for a presence or absence of red colored spore inside colorless vegetative cell against a black background. Here, the 10% nigrosin is acts as a decolorizer. Selectively, it decolorizes the color taken by a vegetative cell, and the cytoplasm of cell appears as a colorless. As well as the 10% nigrosin, it is a acidic stain, it gives a color to a background as a black, whereas the spore appears as a red in color. So this is a image of a endospore staining of a streptobacilli. The bacilli are in a chains. The red colored endospore is here, colorless cytoplasm or colorless vegetative cell, and black colored background. This is an overall observation for endospore staining. So thank you for watching me. So please subscribe my channel Easy Life Science, like my video, share it to your friends and press the bell icon.